So today we'll talk about DNA modifying enzymes, and in this lecture we'll discuss how both the insert and the vector can be modified so as to ensure efficient ligation and also to improve the efficiency of transformation and of course the efficiency of DNA amplification or the production of the enzymes. And in the process we'll talk about several different topics which include several enzymes like DNA ligase, alkaline phosphatase and their function, polynucleotide kinase and their function, and how they can be utilized for improving the efficiency of ligation. We'll also talk about terminal nucleotidyl transferase and of course their role in formation of recombinant DNA. We'll also talk about linkers and we'll also talk about adapters. These are double-stranded molecules that can be used to improve the efficiency of ligation for generation of recombinant DNA. And we'll come to each of these topics one by one and see them in details. But before everything else, we start with a bit of a recap of lecture 1. And if you remember, lecture 1 was totally directed to restriction enzymes and their role in generation of recombinant DNA technology. So we begin with the very basics, and then in 1 or 2, 3 slides, we do a bit of a revision and revise the main points of the last lecture. And then we move on and we talk of other enzymes that play a role in DNA modification and ensuring that efficient ligation takes place, which is a key step in actual recombinant DNA technology experiments. So then we move on, and first, of course, if you remember from the previous lectures, we talked of phosphodiester bond. So individual nucleotides on the same DNA strand. So this is one strand here, this is the other strand here. Individual nucleotides in the same DNA strand are linked together by phosphodiester bond. So this is one nucleotide here, this is another nucleotide here, and the two are linked together by phosphodiester bond. A phosphodiester bond is formed between the 3' OH of one nucleotide and the 5' phosphate of the other nucleotide. And there is a loss of a water molecule, and you have the formation of phosphodiester bond. This is to link two nucleotides on the same strand. Right. So this is very important also because if you look at the DNA and if you look at each of the strands, each strand has its orientation. The free phosphate at the 5' end and a free OH at the 3' end. And on the other strand, the orientation is anti-parallel, which means if this has a 5' end here, the free phosphate, it will have a 3' end here with a free OH molecule here. And likewise at the other end, you'll have a 3' OH end here and a 5' phosphate end here. Right? So this is very important. And restriction enzymes, what do? They basically hydrolyze these phosphodiester bonds and therefore break the DNA molecule into fragments. So the phosphodiester bond can be hydrolyzed by restriction endonucleases and formed by ligases. So the other important point in phosphodiester bonds is that the formation of phosphodiester bond happens via ligases, right? And that is again equally important, and we discussed the role of ligases in the earlier lecture, and here again we'll do a bit more detailed discussion on ligases. So here you are. This is your piece of DNA here, a double-stranded DNA with a 5' phosphate and a 3' OH in the top strand, and likewise a 3' OH, which is opposite to the 5' phosphate. Remember, the strands are anti-parallel, and likewise at the other end, because on the top strand there is 3' OH, on the complementary strand you have a 5' phosphate. So the first what you do is you digest this with restriction enzymes, and when you digest this with restriction enzymes, you get two fragments here. So you have an generation of 3' OH and a 5' phosphate at this molecule end here, and a corresponding 5' phosphate and 3' OH on the other molecule here. And then, of course, if you add ligase now to this reaction, because you have a 3' OH and a 5' phosphate adjacent to each other, they would then again form a phosphodiester bond and join together into a single DNA molecule. So this is basically the main enzymes here in recombinant DNA technology, the restriction endonucleases and DNA ligase. Restriction endonucleases are going to hydrolyze the phosphodiester bond, while DNA ligase are going to form the phosphodiester bond. So that is again very important, and the two most critical enzymes in recombinant DNA technology. But these are not exclusively the enzymes that are used here. You have other enzymes to ensure that the efficiency of ligation is high, as also to ensure that there is efficient directional cloning and there is efficient transformation and so on and so forth. And we'll discuss those in this lecture here. Right now, as an activity, I'm asking you a question here. So if a linear DNA has 4 restriction sites and all are digested, how many fragments will be formed? The other part of the question is, would it differ, and if so, how in case of circular DNA? So what you can do is, instead of just straight away answering this, you can draw a diagram, a linear DNA, and introduce 4 cut sites and see how many fragments are formed. And likewise, draw a circular DNA with 4 cut sites and see how many restriction sites are formed. We'll discuss this question in more details when we discuss restriction mapping in module 2. For now, we focus on enzymes that are important and play a role in ensuring efficient ligation, efficient transformation, and also the success of recombinant DNA technology as such. So let's move on, and we talk now of the very basics of recombinant DNA technology again. So here is your vector. In this case, we have a plasmid. Here is a DNA of interest, which we also call as insert, right? Or gene of interest or DNA of interest. Gene of interest is not so much of a correct terminology because this may not essentially be a gene. What you want to do is to ensure that you have a single covalent DNA molecule that has now joined together both the DNAs that you have given in the reaction. So the plasmid should be able to open up and accommodate this piece of DNA or insert into it and then form a closed covalent circle again, right? So this is the whole idea of recombinant DNA technology in its very basic and very simple explanation. So this happens in 2 steps, as I've already told you. So the first step is where you have your step 1, the restriction endonuclease is going to open up the plasmid. And then in step 2, the linearized plasmid or the open plasmid will now take up the insert via ligase and form a recombinant plasmid, right? So this is the basic reaction here. The first, you have the hydrolysis of phosphodiester bonds, opening up the vector, in this case a plasmid, which is a double-stranded closed circular DNA. And then, of course, in step 2, what you're having is basically the addition of the insert into the vector, and then the sealing of the gaps here or the bonds that are broken, so as to ensure that you have a recombinant plasmid here. And this happens via ligase. And I've already told you that the discovery of restriction enzymes was a very fundamental discovery. It provided you a first handle to manipulate the DNA, and therefore Werner Arbor, Daniel Nathans, and Hamilton Smith were awarded the Nobel Prize in 1978 for physiology and medicine. And as I've already told you, it was a fundamental discovery allowing scientists to manipulate DNA and form recombinant DNA using restriction endonucleases and DNA ligase. So this is basically your for revision purpose. And now we move on and we talk a bit more details of this reaction. And then, of course, we talk what are the complications that can come up here and how those complications can be addressed so as to ensure efficient ligation. So if you remember from the previous lecture again, we had discussed about restriction digestion, and I told you there are 2 types of cuts or 2 types of ends that can be generated on digestion by restriction enzymes. You could have what is known as a staggered cut, where there is an unpaired fraction of the DNA on one of the strands. And then you could have what is known as a blunt cut, where there is nothing that is unpaired. The 2 fragments do not have any overhangs or unpaired single-stranded ends. So here in this case, we also emphasize that it is a staggered cut that plays a key role in recombinant DNA technology, and there are several reasons for it. So first, let's see what is a staggered cut. So if you see here, this is the BAM-H1 recognition sequence. It is a 6 base pair sequence, and if you remember also from the previous lecture, the sequence is palindromic, which means it is read the same on top and bottom strand of the DNA, provided it is read from 5' to 3'. So this is GGA TCC, and here again it is GGA TCC. And the other important thing that is important for BAM-H1 is, if you see here, this is the site where the phosphodiester bond is hydrolyzed on the top strand, and here again on the bottom strand, this is the site where the hydrolysis occurs. And very clearly, the 2 sites are not exactly the same position on the top and bottom strand. And when you do such type of digestion, what you generate is 2 fragments which have a bit of unpaired DNA. So this is a 4 base pair overhang that is generated upon digestion by BAM-H1. And this is GATC if you see here, and on the other fragment also it is GATC read from 5' to 3', right? And if you see, because this is coming from what was previously bonded by hydrogen bonds here, the 2 overhangs are complementary to each other. So CG, TA, AT, and GC, right? So they have a natural tendency to come together and get stabilized by hydrogen bond formation. So what happens here is, once you generate these fragments, if there are sticky ends or cohesive ends or overhangs that are complementary, the 2 fragments of DNA will come together and get stabilized by hydrogen bond formation. And this stability provided by hydrogen bond formation because of complementary sticky ends allows a much more efficient ligation by DNA ligase, because now the DNA ligase simply has to come and seal the phosphodiester bond between the GG here and the GG here, right? So therefore, the sticky end ligation is much more efficient as compared to blunt end ligation, because in blunt end ligation, there is no natural affinity between the fragments to come together. And therefore, it is only by a matter of chance that two fragments are too close together that the ligase is able to stitch the phosphodiester bond together, right? So therefore, if you look here, there are advantages of sticky end ligation. One, of course, is higher efficiency of ligation than the blunt end ligation, which we have discussed now. The other important thing is that in many cases, what we also want is that the orientation of the DNA of interest is in a specific orientation, as in, let's say you're doing a cloning for expression of the protein, so exon 1 must come after the promoter, and the exon 4 must come just before the terminated sequence. And therefore, this orientation becomes very important. Such a cloning where the orientation or the direction in which the DNA fragment gets ligated is important to known as directional cloning, and we'll discuss this in more details towards the end of the lecture. In case you have a blunt end insert, and many times what is happening is that your DNA of interest or your gene of interest or your insert is being generated by PCR amplification. So when you do a PCR amplification, obviously what you're going to get is a blunt end DNA molecule. So this blunt end DNA molecule may not very efficiently ligate with its vector. And what we want to do is to ensure that the ligation happens with very high efficiency. And for that, it is important that this blunt end insert is converted into a sticky end insert. And this can be done by several strategies. These include addition of linkers, and we'll discuss what linkers are in the coming slides. Linkers, since they had a certain intrinsic problem, we shifted to adapters, and we'll discuss what adapters are in the coming slides. And then, of course, what we can also do is extend the 3' ends of the insert and the vector with complementary nucleotides so that they have complementary ends to each other. So this process is known as homopolymer tailing, and we'll discuss this as well as we move along. As an activity I have here is give names of two enzymes that leave a same overhang sequence on digestion. So if you remember, I had also asked in the last lecture about looking for isocodimers and isosisomers and new isosisomers. So in addition to that, now what I want you to know also is and search for is two enzymes that leave exactly the same overhangs. So these are basically isocodimers, and please go ahead and do a bit of homework and search for such enzyme names and enzyme pairs that leave exactly same overhangs, right? So let's now have a look at the function of DNA ligase. So here are two DNA molecules. This is molecule 1, this is the other molecule here. And if you see, they are positioned in such a way that the 3' OH of one is in adjacent position to 5' phosphate of the other. Likewise, the 5' phosphate on the bottom strand of one molecule is adjacent to the 3' OH on the other molecule on the other strand. So here in this case, when you introduce DNA ligase, the DNA ligase is going to form a phosphodiester bond here and a phosphodiester bond here. And during the process, the water molecule is going to be released. And therefore, when you put in DNA ligase here, the two molecules join together into a single DNA molecule here. And the phosphodiester bonds I indicate here with the help of these blue circles here. So DNA ligase facilitates the joining of DNA strands together by catalyzing the formation of phosphodiester bonds. So it is antagonistic to restriction enzymes which hydrolyze the phosphodiester bond. DNA ligase, on the other hand, helps in formation of the phosphodiester bond. And then, of course, DNA ligase, because it can form phosphodiester bonds, it is used in cellular machinery in DNA repair. So wherever a phosphodiester bond has been hydrolyzed and a new phosphodiester bond formation is required for repair of the DNA, DNA ligase is brought in. Also, if you remember, during DNA replication, on the lagging strand, the synthesis of DNA happens only in patches. And these small fragments of DNA that are formed are known as Okazaki fragments. And these Okazaki fragments have to be resealed by DNA ligase to get the actual full complementary DNA on the lagging strand. And I'm sure you'll read more about this in your molecular biology classes. So I'll leave it for our molecular biology classes to go into the details. For our purpose here, DNA ligase is meant to form phosphodiester bonds, and that connects the two DNA molecules that may exist that are two different DNA molecules into a single DNA molecule here, right? And as an activity, I want to look into this simple question. So by now, I'm sure you would have read about DNA polymerase as well. So DNA polymerase is basically the chief enzyme that is required for DNA replication. And if you look into DNA polymerase function as well, the major function of DNA polymerase again is to form phosphodiester bond. But there is a subtle difference between the DNA polymerase and DNA ligase. So I want you to check out for that. What is the difference between DNA polymerase and DNA ligase, since both are involved in formation of phosphodiester bonds, if you look at the basic reaction that they're catalyzing. Then we move on and we look into strategies to improve ligation efficiency. And what are the bottlenecks to ligation is the first thing, and then how can you improve the ligation efficiency is the second thing that I want to look into. So here is your basic reaction. This is a reaction that we've seen several times by now. So let's go into it one more time. So here is your linearized vector, and this is your DNA of interest or your insert, and you use ligase, and what you accept if everything is absolutely fine, you should get a recombinant vector, which would be a single double-stranded covalent molecule that would basically also contain your insert and your vector into a single molecule, right? But what actually happens beyond this is also that when you do a ligation reaction, since your vector vector also contains complementary ends, and even if it is a blunt end ligation is possible. So what can also happen is that the vector self-ligates and forms a non-recombinant vector one more time here, right? So in that case, your insert is not inserted into the vector. So this is what happens here. So if you see, alongside the recombinant vector, you also get a self-ligated vector where there is no insert and the vector simply has resealed its phosphodiester bond, forming the same original molecule as it was initially when you open it up. So the strategies now are basically to improve the efficiency of ligation. First, of course, what we want is that there should not be so much of a self-ligation of the vector. And if possible, we want to absolutely eliminate this idea of self-ligation of vector. And also what we want is that the ligation efficiency improves here. And for that, I've already told you that that sticky end ligation is a lot more efficient than blunt end ligation. So therefore, what we most commonly do is a sticky end ligation. If the insert does not have sticky ends, you can generate sticky ends by following certain strategies, which we'll discuss during the course of the lecture. Likewise, if the vector does not have sticky ends, the sticky ends from the vector can be generated. And what we also have to ensure is that the sticky end generated on the insert is complementary to the sticky end on the vector, so that they still have affinity to each other and therefore facilitate your ligation reaction here. So there are two major strategies to improve ligation efficiency. First, of course, is to prevent the self-ligation of plasmid. So you want to ensure that self-ligation of plasmid does not happen, and this can most commonly be done by what is known as dephosphorylation of the plasmid, and we'll discuss that in more details in coming slides. And then, of course, what we want to ensure is that instead of blunt end ligation, what we want to ensure is that you have a sticky end ligation where the molecules have a natural affinity to come to close to each other and form and stabilize the hydrogen bonds, as I've discussed in the previous slides. And then, of course, the job of DNA ligase becomes easy because the molecule is already stabilized. All it needs to do is to form a phosphodiester bond. So key terms here, vector and insert, we've already discussed enough, so we'll not discuss them. Restriction digestion, we have had a complete lecture on this in the previous series. So restriction digestion, I've told you, this is brought about by restriction enzymes. And restriction enzymes can bring about two types of digestion. They can generate sticky ends or they can generate blunt ends. In case of recombinant DNA technology, we prefer sticky ends because they have a natural affinity to come together, and therefore the job of ligase gets a lot easier and ligation efficiency is much higher. Ligation and recombinant DNA, so basically two DNA molecules which may be of independent source, when they're joined together into a single DNA molecule, that is a recombinant DNA. And this joining of the two DNA molecules into a single DNA molecule is brought about by ligase, and the process is known as ligation. So we now move on and we again look at this process in more details and how basically is there a way by which you can prevent self-ligation of vectors is the first thing that we're trying to attempt and understand. So here you are. This is your linearized vector, the plasmid. This is your DNA of interest again, and then of course you use ligase and you get your recombinant plasmid, and alongside you get as a free gift your self-ligated plasmid. And our idea is to ensure that self-ligation of plasmid does not happen. So when the self-ligation is not happening, automatically most of the reaction equilibrium will shift to formation of recombinant plasmid, and therefore the efficiency of ligation would improve. So how do we do that is the question. So let's have a look at this in more details here. So we are zooming in here. This is your recombinant plasmid. This is your insert here in yellow and green, and in pink and blue is your left vector arm and the right vector arm. So here you are. What is the reaction that is taking place here is this. So this is your insert here. This is your left arm. This is your right arm of the vector. And you basically have the formation of a phosphodiester bond here because the 3' OH and 5' phosphate are adjacent to each other on two different DNA molecules, and therefore it is very easy to form a phosphodiester bond by ligase. And if it is a sticky end and complementary sticky ends, it becomes all the more easy for ligase to form a phosphodiester bond. So this is what is happening at the recombinant plasmid here. The phosphodiester bond formation takes place at this point, and likewise in the bottom strand here and here. So there are 4 phosphodiester bonds that are formed, resulting in formation of a single covalently linked DNA molecule here. Now let's look at what is happening at the vector end. So here is your vector, and this was where the cut was introduced, and now this is where the cut has been sealed. So at the vector end also what is happening is that this is the left arm, this is the right arm, and the left arm and right arm are so positioned that the 3' OH and 5' phosphate are adjacent to each other, and therefore the ligase can come in and form a phosphodiester bond here and a phosphodiester bond here, forming your self-ligated plasmid, which is this. And this is something that we do not want in our reaction actually, right? And the question then is, can we prevent the self-ligation of the vector? And alongside, I also want to ask you for a certain activity here. So again, in biotechnology, in every experiment, what we want to do is to check whether the current objective has been achieved before moving on to the next one. So after ligation, you move to transformation, but before that, you need to check whether a ligation has happened and whether you have more of the recombinant plasmid as compared to self-ligated plasmid or not. So a very common way of checking how much of the recombinant plasmid is present in a ligation mix is to do a PCR, right? And I'm sure you would have read about the PCR or polymerase chain reaction. So in PCR, we have primers. So you have a primer here. This is inside the vector and facing towards the insert. And likewise, you have the right primer here again on the vector and facing towards the insert. And then, of course, you do an amplification. So when you amplify, entire region, which includes the insert, will be amplified here. In case of self-ligated plasmid here, your primer is here and here again. So because there is nothing else in between, so only the vector region will be amplified. So this can give you a different difference in sizes of the bands that you obtained on a recombinant plasmid and a self-ligated plasmid. So here is the question for you. If the PCR primers to check the ligation are 100 base pair apart in the vector sequence and the insert size is 400 bases, then what will be the observed band sizes for recombinant and self-ligated plasmid after PCR of the ligation mix? So, and some of you may be able to answer this question right away, and some of you who have not understood PCR correctly or who have not read PCR till now, well, of course, for them, I can assure you that there will be a lecture on PCR in the series itself. I think it is a topic in module 2, and there we'll be discussing in absolute details how a PCR works. And also I'll give you practical examples of what would be a solution to this problem here, right? So this is basically what we do. And now coming back to the original question, that is, can we prevent self-ligation of the vector? So the answer to this is alkaline phosphatase. So alkaline phosphatase basically removes the 5' phosphate from nucleic acid oligos. So here is a DNA molecule with 5' phosphate on the top strand and at the opposite on the bottom strand. Remember, the two strands are anti-parallel. So then when you use alkaline phosphatase, you have the loss of 5' phosphate here and here. And alkaline phosphatases are used basically to remove 5' phosphate from DNA molecules, and this serves in dual purposes. So first, of course, we remove the 5' phosphate from vector to ensure that self-ligation of the vector does not occur. How this works out, we'll see in details in the next slide. And because the self-ligation of the vector does not happen, this also facilitates the ligation of the vector with the insert, right? And then, of course, the other important area where this is used is basically in in the hybridization experiments, there are two types of hybridization experiments based on the substrate that you use. So you have southern hybridization and you have northern hybridization. And in these cases, what you use is to basically generate a probe. A probe is an oligonucleotide that contains a radio label either at the terminal ends or all along, right? And most commonly what we do is we use a probe that is at the terminal end. And in that case, what you have to do is to basically remove the normal phosphate using alkaline phosphatase and then add radio labeled phosphate to the end. And that is done by polynucleotide kinase, right? So this is the basic idea of southern and northern hybridization. And of course, you'll have a lecture on this as well in module 2, where we'll discuss in details the basic principles of southern and northern hybridization, as also the applications of southern and northern hybridization. So as an activity, I want you to look up here again, and I've already given you the answer to this question just a while ago. So the question is, which enzyme performs function antagonistic to alkaline phosphatase? So now we look into how use of alkaline phosphatase allows you to prevent self-ligation of vector and facilitates the ligation of vector and the insert, right? So here you are. This is your vector, and this vector has been first opened up by using restriction enzyme, and then of course it has been dephosphorylated using alkaline phosphatase. So a vector may be opened and dephosphorylated to ensure that no self-ligation takes place. Facilitates ligation between vector and insert. So the question is, why should the ligation not happen here between the vector ends now? So this is the question that we're going to answer now. So here is your vector ends. This is your left arm. This one is your left arm here. This is your left arm. Here is your right arm. And if you can see, because they have been treated with alkaline phosphatase, now there is no 5' phosphate. So because there is no 5' phosphate, this is on the either vector arm here. Therefore, what happens is there cannot be a formation of phosphodiester bond. For the formation of a phosphodiester bond by ligase, you must have a 3' OH and you must have a 5' phosphate. Because there is no 5' phosphate present, now the two ends cannot ligate to each other. So therefore, vector self-ligation does not happen. Now let's see how the ligation happens between the insert and the vector now, right? So here is the other idea. So first, of course, self-ligation is not possible since the 5' phosphate and the 3' OH ends are not present adjacent to each other, as the 5' ends are dephosphorylated by alkaline phosphatase. Now let's look at how this aligns with the insert and how the ligation can happen between the insert and the vector. So here is your insert, and insert has the 5' phosphate intact because it has not been treated with alkaline phosphatase. And here is how your vector arms are going to align with the insert. So this is the left arm here, this is the right arm here. And if you can see at two positions, there is a 5' phosphate and a 3' OH next to each other. So therefore, here your phosphodiester bond formation will take place. One position is here, the other position is here, where you have again a 5' phosphate and a 3' OH. So 3' OH is coming from the vector and the 5' phosphate is coming from the insert. And here again, you'll have the formation of a phosphodiester bond. On the other two positions where normally you would have a phosphodiester bond, because we do not have a 5' phosphate here and a 5' phosphate here is missing. Therefore, no phosphodiester bond formation takes place at these positions. So these remain as unconnected nucleotides with each other. So this is basically a neck that you have here. So the two strands are complementary to each other. Therefore, the actual recombinant DNA is still stable. So base complementarity keeps the recombinant DNA stable despite necks. And then, of course, what happens is the necks are basically repaired after transformation using the host cell machine, right? So as I told you, blunt end ligation is very inefficient, and this is more so in case where the two molecules to be ligated are large. So for example, you have a 500 base pair insert and you have a 3000 or 4000 base pair plasmid. So in such cases, again, blunt end ligation becomes slightly difficult, right? And it is highly inefficient. In contrast, sticky end ligation is 10 to 100 times more efficient than blunt end ligation. So an effort is always made to ensure that your insert and your vector have sticky ends. And there are several ways by which you can ensure that your insert has a sticky end. Let's say your insert is coming from a PCR amplification. So in such cases, the insert will not have a sticky end. And therefore, sticky ends need to be added to the insert to ensure that the ligation is efficient. And there are several strategies for doing that. The three most common strategies that are used to generate sticky ends to blunt end inserts include linkers, adapters, and homopolymer tailing. And we'll now go into the details of each one of these as we move along. So we start with linkers first, and then we move on to adapters and also to homopolymer tailing. So coming back to linkers now. So linkers, this is basically, if you look at linkers have 16 to 20 base complementary synthetic oligos with blunt ends. So this is a linker here. If you see, this is a 16 base pair linker. You have 5 bases on this side, 6 bases of recognition sequence, another 5 bases of extra sequence on the 3' on the right hand side, and together this is a 16 base pair sequence. And if you see very clearly, in the center of which you have a recognition sequence for BAM-H1. So this is a BAM-H1 linker. Linkers are 16 to 20 base complementary synthetic oligos with blunt ends, and they contain, within the sequence, they contain a unique recognition sequence for one enzyme. In this case, this is a BAM-H1 recognition sequence. It has the recognition sequence for BAM-H1, which is GGA-TCC, right? So this is a 6 base sequence here. And if you see, this is a palindrome again, as has been repeated many times in the lectures now. The recognition sequence are palindromic in nature, which means they can be read the same when read from 5' to 3' on both the strands. So this is GGA-TCC, and here again it is GGA-TCC, right? So this is your linker here, and what you do then is you set up a ligation reaction with your insert and the linkers along with it, and this is what you get on the ligation reaction. So here is your linker. I represent this with the red representing the recognition sequence and the black representing the extra nucleotides on the sides that you have to make the complete linker here, right? And now when you do a ligation reaction, so you have your insert plus linkers and plus DNA ligase, what you get is a molecule where insert gets attached to linkers and linkers get attached to themselves, forming a large macromolecule here. So this is your insert, and this is attached to your linker molecules, which are then attached to each other in multiple copies. And after you've done a ligation reaction, you digest this with BAM-H1, and on digestion with BAM-H1, now you'll have basically the generation of an insert with BAM-H1 sticky end. So this is the CTAG overhang at this end. At the other end also, you have a CTG overhang on the top strand. So this is basically your insert now with sticky ends, and this insert can be taken forward for the ligation and transformation reactions. If you zoom in into this region here, this is basically the overhang that you have for BAM-H1 linkers. So if you see, this is the common region here, the black one, and then of course the overhang that generates after the digestion by restriction enzyme BAM-H1. So you have a CTAG overhang at this end. Likewise, the same molecule in the converse orientation here. So you have a CTG overhang on the top strand, and you have a CTG overhang at the bottom strand here. So this is your insert molecule now, which has been converted from blunt end to sticky end and can be taken forward for ligation and for transformation. And ligation efficiency would improve, as I've already told you, because it has sticky ends now. Now, there is one activity that I want you to think about. So can you think of a possible problem that can come with a linker? And let me also put it in context. Linkers were used at a time when there was not enough sequence information. And let's say also, if you have this insert that you are trying to clone for sequencing, so which means obviously you do not know the sequence of the insert. So given this context, can you think of a possible problem that can come up with linker, given that we are using a random recognition sequence linker to be attached to it and then digest it with the same recognition restriction enzyme? So I'll give you probably 30 seconds to think over it, and then of course we move on and we see the solution to this. So here are your 30 seconds now. Your timing starts now. 4, 5, 6, 7, 8, 9, 10. So this is your half minute, and I'm sure you would have got the answer, but in case you haven't, so the problem is that we are not sure whether the BAM-H1 recognition sequence is present inside our insert or not. If it is not present, then there is not a problem. But if it is present, then of course your insert itself gets digested. And what you get is something like this here. So here is your insert, and let's say your insert also had a BAM-H1 recognition sequence inside it. So when you do a digestion with BAM-H1 after ligation with BAM-H1 linkers, your insert itself would get digested at the site where the recognition sequence is present inside the insert. So therefore, the entire purpose of trying to clone this gene DNA of interest into a cell is going to be defeated, because your insert is already broken in between or disrupted in between. So therefore, linkers were a bit of a problem, and people moved on to what is known as adapters, right? And in adapters, we discuss more about adapters in the next slide. Adapters are partially double-stranded synthetic molecules with one blunt end and one sticky end. And the other important property of adapters is that at the sticky end, they lack a 5' phosphate. So this is a typical adapter here. You can see this is partially double-stranded, and you have basically the blunt end that is here, and 5' phosphate is present at the blunt end, and at the sticky end, the 5' phosphate is not there, right? So the 5' phosphate is absent at the sticky end, and this is important because this serves to prevent adapter self-ligation. And how does it happen is what I'm going to show you next here. So why lack of phosphate at sticky ends is important is because if it was present, then what you would have is adapter self-ligation. So here is one adapter here, there is the other adapter molecule in the opposite orientation, and you can see very clearly the overhangs are complementary, and they can very easily form hydrogen bonds. And once this molecule is stabilized by hydrogen bonds, DNA ligase can very easily come in and form the phosphodiester bond. So this is a sticky end ligation that would happen, and because the ends are complementary, it becomes all the more easy to form phosphodiester bonds. And therefore, to avoid this condition where the presence of a phosphate at the sticky end allows for adapter self-ligation, you do not have a phosphate at the 5' end of the sticky end of the adapter. So let's now look at how the interaction between adapter and the insert happens. So here is your blunt end insert, and these are our adapter molecules, one at this end and the other at this end. And you know that adapters cannot ligate through their sticky end, because at the sticky end, there is no 5' phosphate. So only ligation that happens is at the blunt end, through the blunt end between the insert and the adapter here. And likewise, the ligation reaction can happen here. Alongside, you can also have some molecules where the adapters are ligated to each other through the blunt end. So now finally, from a blunt end insert, after the ligation of the adapters, what you get is a sticky end insert, and this sticky end insert already has sticky ends because the sticky ends are inbuilt into the adapters, right? So now you can take this molecule forward for your next steps of ligation, and then subsequent ligation with the vector, and subsequent to that, transformation. But then there's a catch here. If you remember here, the adapters do not have a 5' phosphate at the sticky end. So that has to be restored before you can move forward with the ligation and transformation downstream. So let's see how this addition of phosphate groups happens on the sticky end insert that now has adapters. So here you are. This is your insert with the sticky end adapters. And if you zoom in into this end of the adapter here, so this is where you're zooming in. So here is the insert part. This is where the blunt end ligation has taken place, and this is the adapter part. And if you see, the sticky end of the adapter does not have a 5' phosphate. So this 5' phosphate has to be restored here, so that now these ends become okay for ligation with the vector. So what you do here is, so adapters, after the adapters have been ligated, the insert lacks phosphorylated 5' ends. The 5' phosphate is restored by treatment with polynucleotide kinase. So the enzyme that we use here is polynucleotide kinase, and polynucleotide kinase in the presence of DATPs. So DATP is the donor of the phosphate group here, will allow for the addition of phosphate group at the 5' end of your sticky end of the adapter here. So now, once you have done this, at both the overhangs, the 5' phosphate is restored. This molecule is now ready for your ligation with the vector here. So the insert can now be ligated to the vector. Now coming to the third method that can be used to ensure that you have generated sticky ends and complementary sticky ends. Both the insert and the vector is what is known as homopolymer tailing, right? And I'm sure you would have heard of many polymerases till now. And most commonly, of course, you would have heard of DNA polymerase and RNA polymerase. And you know that there are several types of DNA polymerases, and there could also be different types of RNA polymerases in eukaryotes. So what happens is these DNA polymerase and RNA polymerase are actually template dependent. So they add nucleotides complementary to what is present on the template, right? So these are template dependent polymerases. In contrast, terminal deoxynucleotidyl transferase is template independent. So it can independently attach nucleotides to the 3' OH end of a pre-existing oligonucleotide, provided the DNTPs are fed into the system, right? And here in this case, I show you the example of how you're going to modify your insert and the vector so as to generate complementary sticky ends. So this is your terminal transferase. Terminal transferase or terminal deoxynucleotidyl transferase when it is working on DNA. So terminal transferase are template independent. They add, terminal transferase can add DNTPs at the 3' end of nucleic acids, and they do not require a template for this. So if you give one kind of DNTP only, several such DNTPs would get added to each other, giving you a homopolymer tail, right? Homopolymer tail means essentially the same nucleotide is getting added multiple times. So here, if you see now, this is your insert. So here is your 5' phosphate on the bottom strand, 5' phosphate on the top strand, and this is your 3' OH end on bottom and top strands here. And terminal transfer is going to work at the 3' end, and it is going to extend the 3' ends when you give it the DNTPs. So what we very smartly do is we give only one type of DNTP here. So for example, here in this case, for the insert, we're giving only DCTPs, which means only the cytosine nucleotides are being given. So upon in the presence of terminal transferase, you will have the addition of multiple cytosines at the 3' OH on both the strands of the DNA here, right? And likewise, what we do is very smartly. Also, what we do is for the vector now, we extend the vector using DGTP. So G and C, are complementary base pairs. So therefore, that becomes that would help you generate the sticky ends. So here I show you the vector arms here, the left arm and the right arm. So here you have your vector left and right arms, and you can see there is a 3' OH here and a 3' OH here, and you are giving complementary to cytosine. You are giving DGTPs here. And when you give that, now this end, the 3' end of the vector arms is going to be lengthened and increase in length because of the addition of guanine nucleotides. So when you do this reaction, what you get is this, where you have a poly G or homopolymer tail of guanines at the 3' ends of the vector arms here. So this is your guanine here, GN. N means N could be any number depending on how much reaction time you give. The more reaction time you give, the more number of guanines will get added. So this is again very important, and this helps you generate a sticky ends which are complementary to each other on the insert and on the vector at the 3' ends. Some of the key terms here, and I'm sure some of the terms you may not have heard before. So template dependent polymerases. So both DNA polymerase or RNA polymerase are template dependent. Unless there is a template present, they are not able to add nucleotides because they require a template to add complementary nucleotides to a pre-existing oligo. Template independent polymerases, as I told you, so terminal deoxynucleotidyl transferase or terminal transferases are template independent. They can add nucleotides at the 3' end independent of a template, means you do not require a template to be present. If you give a mixture of DNTPs randomly, a certain order of DNTPs will be added to the 3' end. And in this case, in our case, what we do very smartly is we give only one type of DNTP for the insert, and we give a complementary DNTP in the vector. And most commonly it is C and G, because you know that C and G have triple hydrogen bond, and so they are more stable as compared to A and T, and therefore the the initial bonding between the vector and insert would happen through these complementary C and G nucleotides. And because they'll have three triple hydrogen bonds and multiple C's and G's, you will have a more stable molecule to begin with. And then, of course, the ligase can come in and form the phosphodiester bond. And then, of course, we talked about homopolymer tailing. So this is called homopolymer tailing, where you are adding the same nucleotide multiple times at the tail of the DNA molecule. So 3' end of the DNA molecule is generally the tail, and the 5' is basically the head. And remember, the sequence is always written from 5' to 3'. So this is what is known as homopolymer tailing. And let me also then take you to how this actually works and what complications can we get here. So here is what I show you after the extension by terminal transferase. This is what we get. So you get 6 cytosins here and 6 cytosins on the 3' OH ends of the insert, and likewise you get 6 guanine nucleotides at the 3' end of the vector here, right? So there is no problem. You have complementary nucleotides and they are equivalent in number. So these would then join together by hydrogen bonds first, and then of course the ligase would come and form the phosphodiester bonds, finally stabilizing the molecule and making a single DNA molecule of it. So this is basically, these dots represent the phosphodiester bonds that are getting formed at these 4 positions. And this results in finally your recombinant DNA, where the insert is now covalently linked to the vector sequence and basically forms the recombinant DNA. But what can also happen is, because we do not have any control on how many C's and how many G's can get added, so there can be times where you can have a mismatch in the number of G's and the number of C's that have been added. So here I show you in the insert you have 4 C's at each of the 3' OH end, and in the vector you have 6 G's at each of the 3' ends. So now there is a problem in the sense that there is a gap that comes in when they align with each other. So there is a 2 base pair gap here, there's a 2 base pair gap here, or a nick that we call it here. So now what happens is, unless this nick is sealed, you will not be having a phosphodiester bond formation. So this nick is sealed by an enzyme known as klenopolymerase. So, and once the klenopolymerase is used, the klenopolymerase, as it is a polymerase, it will use the template here, and it sees there is a G's on the other strand, so it will add complementary cytosins and 2 in number to ensure that this gap is filled here. Likewise, you'll have the filling of gap here. Again, 2 more C's will be added, and then of course accordingly the 3' OH end will keep moving forward. And after this is done, of course, now you have basically the filling of the gap. So therefore now the ligase can come in and form the phosphodiester bond, sealing the molecule together completely, right? So this is what happens when you use klenopolymerase, and you also along with DNA ligase, right? So this is what is normally done in case of homopolymer tailing. So homopolymer tailing is essentially to generate sticky ends, and generally it is the same nucleotide that is coming multiple times at the 3' end of the insert and the 3' end of the vector, and you very intelligently introduce complementary nucleotides so that the sticky ends generated in the insert and the vector are complementary to each other, and therefore they can allow for easy formation of phosphodiester bonds. In case there is a mismatch in the number of cytosins and guanines that are added, or the number of nucleotides in the vector and the insert at the 3' OH end that you've added using terminal transferase, then in that case you can use klenopolymerase to first fill the gap, and once the gap has been filled, you can then basically join them together using DNA ligase. So some key terms that we have talked of here. So template independent polymerase, I've already told you RNA and RNA polymerase and DNA polymerase are template dependent polymerases. Then template independent polymerase is terminal transferase or terminal deoxynucleotidyl transferase. Then of course we've talked of terminal transferase in more details. What I want you to look for is, what is klenopolymerase, how is it generated, and how are its properties slightly different from the DNA polymerase that we normally use. And this is basically coming from DNA polymerase 1. So how is klenopolymerase generated from DNA polymerase 1, and what are its properties, and why specifically we use klenopolymerase here and not DNA polymerase 1 itself? So these are some of the questions that I wish you to look for an answer in case you don't get it. In some of the lectures downstream, we'll be coming back to it and referring to it again, and there I'll give you more explanation for this. All right, so this was your homopolymer tailing. So we have discussed 3 strategies of how these can be used to generate sticky ends on a blunt end insert, right? So these include your linkers, adapters, and finally your homopolymer tailing. So let's now talk of directional cloning. So if you see here, what we're doing in this experiment is we're trying to express our protein of interest using this insert, which has exon 1 at the start and exon 4, which is the last exon at the end here, right? And what we want to ensure is that exon 1 comes and sits next to the promoter, and exon 4, or the last exon, comes and sits immediately upstream of the terminator sequence on the vector here. So therefore, what we want is that the insert should get attached to the vector in this orientation, where exon 1 is downstream of promoter, immediately downstream of the promoter sequence, and exon 4, the last exon, is immediately upstream of the terminator sequence. However, what can also happen is that the insert can flip over, and it could get inserted in this direction, where E4 comes in just next to the promoter and E1 comes next to the terminator. So this is an orientation that we do not want, because here our protein will not be expressed. So therefore, we have to have mechanisms by which you can avoid this type of orientation of insertion, and ensure that the insertion happens only in the right orientation, which is where the first exon of the coding sequence comes next to the promoter, and the last exon of the coding sequence comes immediately upstream of the terminator sequence in the final recombined vector that we have here, right? So how can we ensure this is a question? So in certain cases, specifically when the insert is being cloned for protein expression, the orientation in which the insert ligates in the vector becomes crucial. Direction cloning is ligating an insert in a specific and known orientation in a vector, and directional cloning can be used, can be ensured if the insert has 2 different sticky ends and the vector has corresponding complementary ends. So what we want to do now is to ensure that our insert has 2 different sticky ends, and the vector is also cut accordingly so as to ensure that it has complementary sticky ends. So let's see how this is done here. So, and before this, there's a small question that I want to ask you, and the question is, and of course we're trying to address this in the next slide, can sticky ends be utilized to force directional cloning? So can we use sticky ends in a manner that only the direction cloning happens, and the other type of cloning where the orientation is reversed to the what is wanted does not happen? So let's see that here. So here is what we're looking at. So again, you have your DNA of interest, and this time it is basically the complementary DNA, which means you have removed the introns here. So you have the exon 1, exon 2, exon 3, and finally the last exon 4 here. So this is your DNA of interest. You want to clone it into this vector here. You want to insert it into this vector here, so that you finally get a recombinant vector that can be used to express the protein that is getting expressed from this gene here. So what you want to do is to get this recombinant vector where the orientation of the coding sequences exactly in the sense that the first exon is next to the promoter, and the last exon is immediately upstream of the terminator. And of course, as I said, you want to avoid the situation where the insert is flipped and you have the orientation of the exons exactly opposite to what you actually want here, right? So therefore, we need to ensure that this does not happen and only this happens. So what you do here is you prepare an insert that has 2 different sticky ends. It is not the same sticky end at both ends. So let's say this is Bametron, then this is let's say Hind3. So you have a Bametron overhang here and a Hind3 overhang here, and accordingly you cut your vector again, and vector also has a Bametron insert overhang here and a Hind3 overhang here. So now this vector and insert combination can only happen in this orientation here. So if you see here, this is the only orientation in which the ligation can happen, because if this flips over, then this is not complementary to this overhang here. Likewise, this Hind3 overhang is not complementary to the Bametron overhang. So therefore, The only orientation in which the two can bind stably and then allow the ligase to form the phosphodiester bond is this orientation, where both the overhangs correspond to each other, and therefore this allows for directional cloning a lot easier as compared to and this favors this type of vector insert interaction. So this is what is your directional cloning. Directional cloning is important for expression of proteins, as I've already told you. And of course, some of the key terms that we've used here. So you have expression vector. So for expressing a protein, you need special vectors that are known as expression vectors. Expression vectors would have a promoter sequence before the MCS or multiple cloning site. We'll talk of what is multiple cloning site in the next lecture, where we'll start with vectors now, and we talk first of plasmid. And there, of course, I'll give you the key concepts of what a vector can contain. And one of the important properties of a vector is to contain what is known as a multiple cloning site. So this is basically a sequence, a stretch of sequence where multiple recognition sequences for different restriction enzymes are present. And we'll talk more of it in the next lecture. So expression vector, just at the start of the MCS, 5' to the MCS, you'll have the promoter sequence, and 3' to the MCS, you'll have a terminator sequence. So these facilitate the expression of the protein. You know, the promoter is the region where the RNA polymerase is going to come and bind and start the transcription. And once the transcription is done, you'll have the translation and expression of your protein. Then, of course, terminator is basically the signal sequence that allows for termination of transcription. Then, of course, we have talked of directional cloning. And I told you, for protein expression, it is important that you have the insertion of the insert in a specific orientation. So this process is known as directional cloning, and this can be facilitated by using two different sets of sticky ends on the insert here, and accordingly you have the complementary sticky ends on the vector here. So this is basically what you get.